In recent years, as the microbiome, and in particular the vaginal microbiome, has risen in awareness, prominence, and scientific curiosity, one particular species of vaginal bacteria has become a sort of darling of this new research space, Lactobacillus crispatus. This bacteria is the most in-demand bacteria in the vaginal microbiome, in part because studies suggest it is protective against all sorts of women's health issues, from preterm birth to bacterial vaginosis to HPV. The vaginal microbiome is a microhabitat of bacteria, fungi, and viruses that occupy every woman's vaginal canal and influence many parts of her reproductive health. Bacteria are only one important part of it, and as noted on a previous episode with Dr. Lynn Sykes from December 2023, the bacteria in the vaginal microbiome can be separated into three groups: the good, the bad, and the ugly. The bad and the ugly are known to cause inflammation, and in the case of pregnancy, to trigger preterm birth. The good, made up of many different types of lactobacillus species, protect against preterm birth and represent the healthiest and most resilient vaginal habitats. But why? What makes this bacteria family, and in particular the Lactobacillus crispatus species, so protective? That's exactly what scientists at the March of Dimes Prematurity Research Center at Imperial College London have been asking. It's a hard scientific question, but not one out of reach for this team, one of the top groups globally, adding to the fascinating science of this emerging field. And very recently, the March of Dimes Imperial PRC team published a paper with the answer to the question, why? Lactobacillus crispatus is good, they found, because it is literally coated in a protective shield that prevents it from latching to pro-inflammatory receptors in the vaginal canal, a type of body armor, an advantage that the bad and ugly bacterial families do not possess, in a class of its own. Plus, their paper found that this protective layer could be detected in a simple vaginal swab, and that its presence was linked with decreased inflammation. What does this discovery mean, and how can scientists use it to delay or prevent preterm birth? That's all ahead. Stay with us.
The views expressed on this podcast may not represent those of March of Dimes or its staff. Information presented on this podcast is not medical advice and should not be construed as such. When in doubt, seek competent medical advice. Now, on with the show. The conversation you're about to hear is about a paper that is currently under publication review. You can view it as a preprint in the notes. This episode is about the vaginal microbiome, preterm birth, and the little bacterial species that could. Let's meet our guests, the authors of the paper that defines the virtue of the bacterial species of the moment, Lactobacillus crispatus. Dr. Alexiane Decout is assistant professor in immunology at the University of Warwick and a research collaborator at Imperial College London. Dr. David MacIntyre is professor of reproductive systems medicine at Imperial College London, one of the directors of the March of Dimes Imperial PRC, and the new director of the Robinson Research Institute at the University of Adelaide. Alexiane joins me from England, and David joins me from Australia. Alexiane, welcome to the show.
Thank you for having me.
David, it's very nice to have you back on, albeit from a different country.
Correct. It's great to be here again speaking to you.
Congratulations on this paper, the both of you and the rest of the team members that were authors on this paper. Let's start with a few basic questions. David, where is the vaginal microbiome exactly? I've often wondered this. Is it inside the vaginal canal? Is it in the uterus? Is it on the labia, the vulva in general, all of the above? Where does this thing reside?
So most of the anatomical locations you've mentioned, they all do have a microbiome, but the composition or the collection of the microbes at each of those body sites that you've mentioned are a little bit different. So for example, the microbes that are colonizing the vulva or the external genitalia, they tend to include some common skin microbes and bacteria. But when most people are talking about the vaginal microbiome, they're really talking about the inside of the vaginal canal. So once you go beyond that, you're reaching the cervix, and beyond that, you're into the uterus, where there tends to be far fewer microbes present. But we do see evidence for colonization of the endometrium or the lining of the uterus in some women.
All right. Okay. So the canal, mostly the canal. Okay, good. And David, I know there's still a lot we don't know, but what do we know about the type of bacteria that colonize this vaginal canal, the vaginal microbiome, and why? Uh, and why do different women have different bacteria in there?
Yeah, you're right. We're still learning a lot about the different types of bacteria that colonize the vaginal microbiome, but we do know that broadly, most women are colonized by one of a few species of bacteria called Lactobacillus. Most commonly, women will have Lactobacillus crispatus or a different species of Lactobacillus called Lactobacillus iners. But there are a couple of other Lactobacillus species that are pretty common in the vagina. Now, women who are colonized by one of these lactobacillus species will typically not have any other bacteria as part of their vaginal microbiome. So, compared to other body sites like the gut or the skin, the vaginal microbiome generally in most women is quite a simple microbiome composition. There are some women, however, who don't have any of these lactobacillus species present, and these women will instead tend to be colonized by lots of different types of bacteria. So they'll have a higher diversity composition. Now, it's important to probably point out at this stage that the compositions of microbes in the vagina are not fixed for life. They are quite dynamic, they're changing. There are several different factors that will influence whether or not you might have one type of bacteria versus another. So, for example, menstruation or sexual intercourse, taking a course of antibiotics, even some hygiene practices are all examples of things that could disturb the microbial communities in the vagina. The reason why we care about this so much is because we and others have shown that there are strong associations and links between specific microbial communities in the vagina and reproductive health outcomes. And you've touched upon some of these in your introduction. We know that women, for example, with higher diversity vaginal microbiomes lacking in lactobacillus species tend to be at higher risk of acquiring sexually transmitted infections, or during pregnancy, they're at higher risk of preterm birth. So we're now focusing a lot of our attention on understanding the mechanisms that are explaining these relationships, and that's what we're going to be talking about today.
Yeah. So I was wrong then in my introduction when I said that it is the most in-demand species. You said that the majority of women actually have lactobacillus, and is it the majority of that majority that has crispatus? So this is kind of the most common. This is a good guy, and a lot of women have this good guy.
Yeah, so there are a lot of women who are carrying Lactobacillus crispatus, but the proportions differ in the different parts of the world. So, for example, I would say in Northern America and Europe, based on the data we have, probably about 30 to 40% of all Caucasian women who are of reproductive age have their vaginal microbiome dominated by Lactobacillus crispatus. But it's less common to find that species in the vagina of African women or American African women. So it is a common species, but it is really the relative abundances of that species that we think are important. When you have a lot of Lactobacillus crispatus, that tends to be associated with healthy outcomes and healthy reproductive outcomes. The other Lactobacillus species don't seem to have the potential to protect against infections and other things that might lead to poorer reproductive health outcomes, which we'll dig into soon.
Yeah. Very quick question. You mentioned reproductive age. Is this bacterial family and this specific species, Crispatus, and all this bacteria, is it specific to the reproductive phase in a woman's life? Or, for example, in menopause, do women have this species in menopause? Do they have it before they start menstruating as young girls? What is the lifespan of this bacteria?
Yeah, that's a really good question. So the relative amounts of these lactobacillus species, including Lactobacillus crispatus, will differ throughout a woman's lifespan. In the early stages of life, when levels of estrogen are quite low, we tend to see women or young girls not being colonized by Lactobacillus species so frequently. As they go through puberty and have an increase in certain hormones like estrogen, which we think are quite important in encouraging Lactobacillus species to grow in the vagina, then you'll start to see an increase in their relative levels. And similarly, at the end of reproductive age, as the menopause is reached, we see lower amounts of estrogen, and in concurrent with that reduction in estrogen, we see lower amounts of Lactobacillus species. So it does change across a lifespan as well.
Got it. So very dependent on estrogen concentrations in the canal and in the hormones that are circulating in the woman. Alexiane, question for you. So obviously this entire episode is about Crispatus. It's about the coating. It's about bacteria. But we also know that inside the microbiome we've got fungi and we've got viruses. We've been, vaginal microbiome research is just focused on the bacteria. From my understanding, I could be wrong. But how important a role do fungi and viruses play in the health? Like, are there also good fungi and good viruses? And which of these three players is the most numerous in the microbiome? Is it bacteria, or are they kind of 33%, 33%, 33%, making up 100% of the different microbiota flora and of the vaginal microbiome?
That's actually a very good point. The research in the field of the vaginal microbiota focused for a long time around mostly bacteria, but indeed fungi and viruses are also part of it. So regarding the fungi, according to the publications that are available, it's approximately 1 in 5 women where fungi are detected in the swabs. So not all women, it's not detected in all women, let's say. However, they are usually correlated with, they have a high impact on health, associated with fungal infections especially. So they may not be present in so many women, but they have a high impact on health. Regarding the viruses, they seem to be present in every woman. Then the consequences of the presence of these viruses is not well described. So it's really a gap in knowledge there. We don't really know what these viruses do. And then obviously the bacteria have been getting the spotlight for most of the time where there has been research on the microbiome. And as David said, obviously we know that Lactobacillus crispatus is associated with health, while other bacteria seems to be associated with pregnancy complications or recurring bacterial infections. So I think all viruses, fungi, and bacteria are important. Bacteria are a lot more studied and very high load. So we have a lot of them. Viruses seems to be there seems to be quite a lot of viruses, but their role is not so well described. And then fungi are not so often present, but when they are highly relevant.
Mm-hmm. Okay, understood. And now, obviously, this is a pretty, like you said, bacteria has been kind of has hogged the spotlight, but the spotlight shining on the bacteria is still relatively new, right? This field is 10, 15 years old, something like this, maybe a little older. When in that time frame, how long ago did researchers like you both and others identify Crispatus as a good guy? How long have we known that this guy is important? This is important for preterm birth and reproductive health and HPV. How long have we known that this is a positive thing?
So Lactobacillus crispatus in itself has been recognized as being a key member of the vaginal community of bacteria for decades, actually.
Okay.
But it is more recently recognized as being strongly associated with health.
Okay.
There are several studies that have shown that this specific species that we're talking about has certain properties and characteristics that allow it to grow very well in the vaginal environment. And we, as I've mentioned, see it mainly recovered from women that do have this sort of optimal vaginal microbiome state. As I've mentioned to you before earlier on, though, the relative amounts of Lactobacillus crispatus that you're finding in different ethnic populations might differ. There's different reasons why it might prefer to grow in certain niches than others. So it's pointing towards there being both genetic and environmental differences, as well as in the way in which the immune system of women have evolved to recognize and respond to different types of Lactobacillus crispatus or strains, which we'll get into, and also other types of bacteria, which are probably shaping overall whether or not we're seeing Lactobacillus crispatus present in certain women and not in others.
Yeah, that's really interesting because, I'm sure a lot of people are saying some women that maybe think that they don't have this as the predominant strain, which again, it's very difficult to tell what you have, right? Currently, there's no way for a woman to know if she has
predominance crispatus or not, she can maybe have an idea that perhaps she has low crispatus or maybe inners, who we'll talk about later, if she has a preterm birth. But there's no way to know. And you certainly, David, I know that you're working on a test that can identify bacteria, see if it is linked with inflammation, if it's causing inflammation, and then you'll know that if this woman is at risk of preterm birth, which is fantastic. And hats off to you and the team for doing that work, because I know that as a woman, I would love to know who I've got, what I have, and the more information, the better for us in the hands of women, certainly. So, and anyway, so what I'm saying is, you just mentioned there are so many different factors that are linked with whether you have crispatus or not. It's not the woman's fault, right? It's not that she's doing something wrong. It has to do with genetics, like you mentioned, other bacteria that are there, all sorts of things. So that's good to know that this is kind of currently out of our reach, but maybe not in the next 5 years. Hopefully there's something we can do about it. Maybe take a suppository or something to flip the flora, as Lynn Sykes is also working on. Okay, so let's talk about inflammation. So this is the big word. What is the relationship between, or what is the difference between crispatus and the bad and ugly guys and their relationship with inflammation, right? Because this is the crux of it. And I'm not asking about the coding just yet, but what is the relationship between this good bacteria and bad bacteria and inflammation, and then all these bad outcomes that can happen?
Yeah, so it's well described in the literature that Lactobacillus crispatus dominans is usually associated with lower level of inflammations, while when you have a high diversity microbiota, then you tend to find in women higher inflammation levels. So that is actually quite well described. There are quite a lot of clinical data around showing that, going in this direction. Now, in our research, we were interested in what were the mechanisms underlying this difference in inflammation caused by crispatus versus the detrimental bacteria, the high diversity microbiota. So the one way the bacteria can promote or decrease inflammation is through interacting with what we call the innate immune system, which is basically the first line of defense of our organisms. And it will detect things that are foreign to us, molecules that shouldn't be in the body and that are suddenly present because of a bug. So that can be beneficial or detrimental. It's just a sensing mechanism. And if you can think of it as a kind of key lock system where our cells expose all these little locks on the surface. And then when the bacteria come, they have a set of keys and they can just activate or not the cells if they have the proper keys on their surface. So that's what we studied. We tried to understand, and that's basically not known in the field of the vaginal microbiota. Nobody has really investigated how the bacteria are detected by our host cells. So that's what we wanted to do. And so our goal is really to understand what are the bacterial molecules, so these keys that interact with our host locks, so receptors, to trigger inflammation or be able to activate the pathways that will shut down inflammation.
Mm-hmm. And before the discovery of this paper, we knew something else about crispatus, right? That it prevents it from causing inflammation. One thing is lactic acid, right? The secretion of lactic acid. Can one of you explain that? And also, is there anything else we knew about crispatus's anti-inflammatory role before the coding discovery?
So we think, yes, lactic acid is produced in high concentrations by Lactobacillus crispatus and other Lactobacillus species. And in the vagina, it does have an anti-inflammatory action. We know that it helps to prevent stimulation of inflammatory pathways, and some of the things that Alexiane has mentioned, we know will be toned down by the presence of high concentrations of lactic acid. But lactic acid also is having another way in contributing to the prevention of the colonization of pathogenic bacteria or viruses. Many of these types of microbes that could be pathogenic in the vagina don't grow well or colonize well in acidic environments. We also think that lactic acid might be playing quite an important role in maintaining a strong epithelial barrier, which is also important in trying to help prevent viruses and viral particles from entering into the bloodstream and causing infections. Lactobacillus crispatus is also quite an incredible bug in that it produces a lot of other compounds that we don't often talk about much. So things like bacteriocins, which are able to target other bacteria and kill them. It also has other techniques and tricks up its sleeve to prevent other bugs from growing. One of them is competitive inhibition. So it really will compete out the different binding sites and the different areas in the vagina that bacteria would otherwise need to reach and adhere and attach to. So it's like many other bacterial species. One of the curious things about Lactobacillus crispatus is that it does actually have a lot of these proteins that Alexiane has mentioned. So embedded into the cell wall that would normally cause the immune system to recognize it and activate an inflammatory response. But as we've talked about, we don't see those types of inflammatory responses associated with Lactobacillus crispatus. And that's really one of the questions that we were trying to address with this paper, is to understand why this bacteria in particular is so good at preventing inflammation and escaping detection, if you like, from the immune components of the host.
Right. The lock and key seem to fit, but they never actually go into each other and cause the inflammation. And the question is why. So let's move on to the paper. The paper found, this is the big finding, this is a perfect transition, David, thank you. The paper found that crispatus has a coating or a layer around it called a surface layer protein, or SLP for short. And this is what prevents the latching, the lock and key from turning, essentially. So can Alexiane or David, can you explain why the layers of protein? I thought the bacteria were topped with sugars and the receptors were topped with proteins, but now we're mentioning that the bacteria also has a surface layer protein. So now I'm confused.
So for the receptor, you are right, they are proteins. So that's not a question.
Okay.
Then the bacteria can actually be covered with a lot of different things, lots of different types of molecules. And in the case of crispatus, the main shield, so this surface layer protein, they are proteins, as the name states, and they will create really a shield around the bacteria. They around like nearly in a pavement-like manner. So they will really cover the entirety of the bacteria and really, protect the bacteria from assault from the outside and prevent detection and prevent the detection of what is below the shield. Now this surface layer protein can have some little decorations, and these are actually sugars. So the surface layer protein, they are protein, but they are also dangling these little sugars that can be detected by the host. And that's what we actually found here. So the
sugars that are dangling from the shield, they are not matching with the receptors, the pro-inflammatory receptors, right? That's what you found.
Not with the pro-inflammatory receptors.
But they do match with one anti-inflammatory receptor.
So what we found is that when we tested a lot of different locks to see if our bacteria was able to interact with these host receptors. And what we found for crispatus was, as David said, that there was no recognition by the inflammatory receptors, which was very highly surprising because the keys are there, we know that. So why is it that it's not detected? And on the anti-inflammatory side, it interacted with one specific anti-inflammatory receptor. And this recognition was mediated by the sugars that were on top of the surface layer proteins.
So you expected the sugars to interact with the proteins on the pro and anti-inflammatory, right? Even knowing what you know about crispatus?
So we didn't expect the sugars on the surface, but the composition of the lactobacilli cell wall has been known for decades, really. And we know that in the lower layers, there are molecules that should be recognized by the host innate immune system, and that actually for crispatus, they clearly aren't recognized. We don't see this interaction at all. And that's really where at the beginning, we were wondering why they were so silent. It was a bit strange because the molecules are there, why aren't they recognized? And it's really because you have this layer of surface layer protein on the surface, this shield, that the host receptors, they can't access the molecule that should trigger inflammation. So there is
Okay,
an
so the
access part.
surprising finding then, Alexiane, was that they were not interacting, and that's when you said, okay, so there's the experiment, the test. You match these guys together, a bunch of different, and we'll get to it, how many bacteria and how many receptors. You matched all of them together, and you were surprised to see that they were not matching with that the Crispatus was not matching with the pro-inflammatory. And then you look deeper as part of the same paper and experiment, and that's when you saw, there's a protective layer. Now it all makes sense. Is that right?
Exactly. Yes, exactly.
Okay. And that was a big aha moment for the two of you?
It was actually, yes. It's always nice when you see things coming together.
I think some of the more curious, surprising things that came out of this too was that we know that other lactobacillus species are not strongly associated with any poor outcomes of reproductive health or anything like that. But we would have thought that they may have also had surface layer proteins in a similar way that are preventing some of these interactions. And that's really what led us to think to ourselves, well, which of these bacteria that have got these types of proteins on their surface, how important are they in modifying the way in which a bacteria is being detected by the immune system and stimulating these sorts of inflammatory pathways? And we were able to really lean on a large collection of bacteria that we have been working up and building on and creating a bank of these types of vaginal bacteria for many years now in our Imperial PRC with some of our collaborators. And so Alexiane was able to dig deeper, to look more carefully at these surface layer proteins, their presence or absence, and what they were doing to the immune receptors in a much more larger scale.
Yeah. So let's talk about the SLP. Who else has the SLP? Nobody? Just Crispatus, or other guys have it too?
So surface layer proteins are proteins. It's a large family that can be found on many different species. Now, if we talk about the vaginal microbiota specifically, the surface layer proteins are found on lactobacilli only. And what is interesting is that it's been described as being present on lactobacilli generally, but in our experiments, we screened several different species of lactobacilli, so Crispatus, Gasseri, Iners, Johnsoni, many different lactobacilli, and we detected consistently only in Lactobacillus crispatus. For the others, there are papers describing the presence on some of surface layer protein on some isolates, but it doesn't seem to be like a general rule. While Lactobacillus crispatus, in our hands, the one that we tested, they always have this surface layer protein present. So it's a very specific feature to have this shield present around the bacteria for the bacteria that we consider as the most beneficial for the vaginal microbiota.
Okay, so the paper, so there's another finding then here. It's not just that this protective shield prevents the latching onto inflammatory receptors and inflammation and all and is protective of all these bad outcomes, but also that consistently Crispatus has this shield, and you can only say that about Crispatus. You cannot say it about any other Lactobacillus strain, and you certainly can't say that about non-Lactobacillus strains. So this is something very special to Crispatus that you see over and over and over again.
Yes, exactly.
Okay, beautiful. And just for folks that are into details, can you talk a little bit about how many bacteria were part of this study and how many receptors were dangled on top, or whether it's the bacteria that were dangled on top of the receptors in order to see who would interact with who? What are the, how many numbers here?
Yes, so we included 17 bacterial species. To cover the range of bacteria that can be found in the vaginal microbiota.
Good, bad, and ugly?
Good, bad, and ugly, all of them.
Okay.
But for most of the bacterial species, we had isolates coming from different women. So that's one of the major points of this study, that we had access, as David said, to this March of Dime collections that was obtained and that was accumulated to have very relevant vaginal bacterial strain that were coming from vaginal samples. So we had in total 45 bacterial isolates, and we tested for Lactobacillus crispatus. We had 11 different Lactobacillus crispatus tested that were coming from different swabs. So I think that's probably one of the largest collection of bacteria that has been tested so far in this type of studies. Now,
Very
regarding
cool. So you said 11 different crispatus coming from different women. So
yeah,
I know it's probably very complicated, but could you try to explain quickly and simply how, for example, your crispatus swab would be different than mine, or how are different? Because I think, oh, crispatus is crispatus is crispatus is crispatus, right? But it sounds like there's variations. And what do those look like?
it's very simple. When we cultivate these bacteria coming from different women, they look different. Some will, when you grow them in solution, some will be very like snowflake-like, they will aggregate. Other will make some very nice homogeneous suspension. They even look different just by eye. So you can tell that if they look different by eye, it means that at a molecular level, you will have differences there. And most likely, they will also not interact in exactly the same way with the host. So it was also something very interesting that we saw that what we consider usually as one homogeneous species, Lactobacillus crispatus, is not that homogeneous, but is actually a range of flavors
Yes.
of crispatus, I would say.
The more you know you don't know. David, you wanted to add something there onto
Yeah,
crispatus, how it's different and unique.
that we know there are actually probably hundreds of different strains of Lactobacillus crispatus. We've obviously been focused on the strains that we've isolated from the vagina, but we actually know that there's other habitats outside of the human body, even that Lactobacillus crispatus can be found in. There's been Lactobacillus crispatus isolated from the gizzards of chickens. Even some plants like olive trees are known to have Lactobacillus crispatus colonizing them. So it's highly likely, we think, based on the data that we've generated in this paper, that some of the strains of very closely related Lactobacillus crispatus strains are likely to be probably better at protecting from infection and preventing inflammation than others are. And that's something else that we think is a key finding of this study. We understand that there is genetic diversity of these different types of very closely related strains, but there's been very few studies that have really looked at whether or not those subtle differences between crispatus isolates are likely to engage with the immune system better than others, or vice versa. And I think that's something we've revealed in this study as well.
Yeah, absolutely. And that's the next step, isn't it? We're going to talk with Alexiane a little bit more about that in a few minutes, and diving deep into the Crispatus family and seeing, whether there are some that are more beneficial than others. I do have a question about the one receptor that the Crispatus did latch to despite the SLP. What is this receptor? What do we know about it? Why this receptor? What do we know about this love affair between these two guys?
Yes, so this receptor, his name is dicicine. It's a receptor that's been known for maybe 30 years. Originally it was identified as interacting with Mycobacterium tuberculosis mostly, and having a role in the tolerance of Mycobacterium tuberculosis in the lung. So we know that it's a receptor that triggers an anti-inflammatory environment that could promote the tolerance of some bacteria. Or if you think about pregnancy, even maybe be beneficial. That's not been shown. I just want to say it, but could promote the tolerance maybe of, could create an environment that would be more favorable to promote tolerance of the fetus, for example. So it's really a receptor that fine tunes the immune environment toward tolerance. And it is also a receptor that recognizes sugars, which is also why we were interested in it as part of the London PRC. What is interesting here is that depending on the sugar, that this receptor can recognize different sugars. And depending on the sugar it recognizes, it will activate slightly different pathways and will lead to slightly different flavors of anti-inflammatory environment. What is interesting is that Lactobacillus crispatus doesn't have any of the usual sugars. So we know it's a sugar that is recognized, but we don't know which one. And we still have to really investigate which type of environment is elicited by this interaction. That's something for the future. But it is very interesting and yeah.
Absolutely. And now what about L inners? I know I want to talk about a little bit about L inners. Now, this is obviously a lactobacillus. Lactobacillus in general are good guys, but this is not a good guy. Would we say that this is a bad guy, or would we say that this is like a guy with a double personality? Like sometimes he's your best friend and other times he stabs you in the back because, he's part of this good family, but actually we see him associated with terrible outcomes, right? We see him associated with, I'm not sure if BV is that one, but certainly preterm birth sometimes, and HPV I know is a big one for inners. So maybe David, can you talk a little bit about this character of L inners and then who did he interact with? What receptors did inners and Gardnerella vaginalis interact with as well during the test?
So I like very much your analogy of L inners being a bit of a split personality character. I think that's very apt. One of the main differences between L inners and Lactobacillus crispatus is the size of their genome or the genetic information that they're carrying. The genome of L inners is about half the size of crispatus, which means it's unable to produce a lot of the enzymes and substrates that would allow it to survive and thrive if given essentially the same food sources as Lactobacillus crispatus would.
Why doesn't it have the same food source?
Well, it will have the same food source, but it won't have the right enzymes or will not be able to make the right enzymes.
I see. So I already like L inners more. I already like him more. It sounds like he wants to be good. He just doesn't have the resources.
So you know what's happened here is I think over time, there's been a relationship that has evolved between Lactobacillus inners and other bacteria that it makes a friendship with, which these other bacteria are producing the types of enzymes and substrates that it actually does need to survive in that community. And so for that reason, we're often seeing women who are colonized with Lactobacillus will have other bacteria co-colonizing in their vagina at the same time. And unfortunately, sometimes the choice of friends of Lactobacillus inners is questionable.
Bad influence.
Exactly. They can end up having a co-colonization with potentially pathogenic bacteria. By itself, L inners as well has been shown to produce some toxins, which are called cytolysins. These can actually create pores or holes in membranes of other cells, like epithelial cells, and that can create damage. But again, that depends on the cell cycle and various other factors, we think. And of course, on top of all of this, our new findings are suggesting that L inners actually might be directly activating some of these receptors and immune responses in the cervical vaginal niche that otherwise wouldn't be activated by L. crispatus. So I'm increasingly convinced that L inners is not just an innocent bystander, and if they're hanging out with the wrong crowd, they might be also doing some bad things.
Yeah. Okay. All right. I'm back to thinking he's a bad guy. Okay. So we know that crispatus doesn't mate with inflammatory receptors. We know that this is a good thing. What about the other guys that do mate with these inflammatory receptors? Is this automatically a bad thing? Does this automatically, or in most cases, mean bad things for reproductive health and preterm birth? Or are there other signals and processes that need to be present in addition to this inflammatory lock and key mechanism for preterm birth to, for the cascade, the inflammatory cascade, as Sykes calls it, to begin, and for preterm birth to happen, or for HPV to linger for years, or for vaginosis to interrupt, to potentially even cause a preterm birth?
Yeah. No, I don't think it's always a bad thing at all.
Okay.
That the key and lock mechanisms are actually activating. In fact, it's probably very, very important that the immune system in pregnancy in particular continues to have the ability to recognize and respond to potentially pathogenic bacteria that could ultimately lead to an infection. In most cases, we think that the immune response is appropriate when you have a pathogenic bacteria appear. It's recognized by these types of interactions that we've been talking about, but that doesn't necessarily lead to any additional complications in the pregnancy. The immune system will deal with it appropriately. But if there is a very strong immune activation that can lead to excessive inflammation in the lower reproductive tract, that definitely can trigger a series of events that could ultimately lead to the premature onset of labor and preterm birth. For example, we know that when you have strong inflammation in and around the cervix, that the cervix is more likely to start to ripen and soften. Inflammation pathways that we're talking about, if they're activated in the amniotic sac or the fetal membranes, that could lead to them to prematurely rupture. And of course, if you activate these same sorts of inflammatory pathways that we're talking about in the muscle part of the uterus called the myometrium, that can actually cause the myometrium to start contracting too soon. So it's really about the timing and the appropriateness of the level of response that the immune system evokes to these bacteria as to whether or not you would turn on a sufficient inflammatory response to drive an early preterm delivery.
Yeah, interesting. And what if there's an inflammatory response in one part of the reproductive tract, like you mentioned, perhaps the cervix is beginning to ripen, but the myometrium inflammatory response is low? Could these two inflammation groups talk to each other and say, "Hey, you're out of line. She's not at 37 weeks yet, or I'm handling whatever is happening here. Don't worry." Like, can they talk to each other and say, "It's not time yet. Let's not get the entire system into go mode, into labor"?
Unfortunately, there is a lot of communication between these systems, but
Yes.
when the process of inflammation starts in one site, it tends to activate pathways that will lead to inflammation happening in other sites. So, for example, if the fetal membranes start to rupture too soon, that will lead to a large increase in compounds called prostaglandins, which are very pro-contractile and pro-inflammatory. And once they're released, that will lead then to the myometrium starting to contract prematurely and the cervix starting to ripen. So unfortunately, you don't tend to see isolated pockets of inflammation within the reproductive tract during pregnancy. Once you have established inflammation in one site, it tends to activate a deeply linked communication pathway between the gestational tissues, which then lead to the onset of labor. So it's really,
Okay, so domino effect.
it is a bit of a domino effect, which is why it's so important throughout pregnancy to maintain a low level of inflammation once the pregnancy is established, until the end of pregnancy, when turning on these pathways of inflammation are necessary. And that's why I mentioned before, the key important thing here is that the timing of the activation of inflammation is appropriate.
So just as a recap then about what we've been talking about, because we're getting to the end of our recording, and I still want to talk about a big piece of news that the paper revealed. But the first thing, just as a recap, is that you identified in this study that Lactobacillus crispatus is not attracting and interacting with inflammatory receptors. This is why it is such a beneficial bacteria to have in the reproductive tract. And you also, the biggest news, identified the reason why this interaction is not happening. It is covered with a coating, a protective shield that prevents it from interacting with these inflammatory receptors. In fact, it only interacts with one good guy, and this is why crispatus is so beneficial and so advantageous. The other really exciting news, maybe even more exciting, certainly equally as exciting, from this paper is that you took vaginal swabs of women and you wanted to see, is this protective layer detectable on the swab? Can we actually see it active and that it exists on the swab, or is some more sophisticated diagnostic tool required? And in fact, you were able to see it on the swab. And not only that, but you tested the swab afterwards, and you saw that SLP was linked with lower inflammation activity in that same swab, right? So tell me about this. This is really exciting, because it's one thing to discover something really cool. It's another thing to realize that it's actually quite an easy application to see whether that is in fact detectable from the woman. So this is very interesting.
Yes, so from what we discussed before, we've only talked about bacteria and what is on the surface of the bacteria. But if you think about the vaginal epithelium, you will have between the host cells, the actual epithelium, and the bacteria, you will have a mucus layer here. And the bacteria tend not to interact too much with the cells because they are physically separated. so that we found if we find something happening on the bacteria that's interesting, now can the bacteria communicate with the host? That's another question. And so that's why we wanted to see if these surface layer protein that are ligands of an anti-inflammatory receptor, as we said before, can they be spread in the environment, shed by the bacteria, and therefore could they possibly cross the mucus and be able to reach the host cells to really have direct contact and an effect? So we didn't know if that was the case. That's why we had a look at vaginal swabs. We looked in the cervicovaginal fluids if we could detect free-floating surface layer protein. And indeed, we could. So that means that the bacteria, they are able to send outside these signals to promote an anti-inflammatory environment, and that this signal will be ultimately able to reach the host and communicate. So that's why it was so interesting for us to see that we were able to detect the surface layer proteins in the cervicovaginal fluids. And then we also had, because we know that it was shed, now did it have an effect? So we had a look at the inflammatory mediators in the cervicovaginal fluid. And what was really interesting to see was that for women who had Crispatus, when the Crispatus was able to shed this surface layer protein in the environment, inflammation was lower than in women who had Crispatus, but Crispatus that were not able to send this signal. So we saw a clear difference between the two. We had a small number of women, but that's really a good indication that some strains of Crispatus are able to send this anti-inflammatory signal in the environment to decrease inflammation, while some other strains may not be able to be so communicative and talk to the maternal environment to decrease a little bit the inflammation. So that was very
Okay, so
interesting.
let's see. And I learned something new here. So this is now whether plot thickens, right? The plot thickens. All Crispatus are not the same. So it's not just that the SLP was detected. See, I thought that the SLP was detected in the swabs, and the women that had the SLP means that they had Crispatus, and it was linked with lower inflammation. And then the ones that was not linked with lower inflammation clearly don't have Crispatus. But that's not what it found at all. It found that the SLPs, some SLPs are able to send this signal, and other SLPs are not able to send the signal. And it is the sending of the signal and the detection of the signal that is linked with lower inflammation. Is that right?
We saw something like, yes, that's what we saw in our sample. However, I must say that we had a small number of samples, so we need obviously to increase our numbers to have something a lot stronger. But
the
yes,
point is that all Crispatus are not behaving the same, right? That's the point. They're not all behaving the same, and they may not all be having this extra positive anti-inflammatory effect. Some of them may have a more dampened anti-inflammatory effect. Is that right?
Yes,
Is
some
that
seems to be
understanding
more
correct?
efficient at triggering,
Yes.
at really dampening the inflammation compared to some other strains. Yes.
Wow. Okay, very interesting. And so then the next steps of this, as David mentioned earlier, and you told me on a background call, is for you to go deeper into the Crispatus strains and see. You mentioned when you look at them in a tube, they look different. Some are fluffier, some are not. Now we know some of them are more efficient at sending these signals than others. So tell me about the next steps of your work together, Alexiane and David, about finding out more about how different Crispatus strains operate and behave, and why this is important. Is it important because of the obvious question? You find the best Crispatus strain and you just kind of manufacture that strain more effectively in the lab, and then perhaps long-term scenario, long ways off, make that into a suppository that a woman can take to flip the flora? Or is it more complicated than that? And is there more involved than I'm letting on?
Yeah. So regarding the immediate work that we will do, there are quite a few things that we need to elucidate following up on this study. The first one is, as I said, we need to increase the number of samples, clinical sample that we have access to, because in our cohort, for
And
example,
how many women, how many swabs did you have, Alexia? And you mentioned not very many, and now you said you need to increase the size. So I'm just wondering, how many did you have, and what sample size would you like ideally in the validation work?
Yes, so we had 56 swabs in total, but only 27 that were dominated by Crispatus.
Okay.
So that's a very small
Lucky
number
girls.
to reach solid conclusion, especially when you want to
Okay.
link inflammation and microbiota.
Okay.
So really, how many we will need, I will need to make power calculation for that. But we need to really increase the numbers. Also because in this 27, we didn't have any preterm birth. So if we want then to associate the presence or absence of the surface layer protein with clinical outcomes, we need to have a significant number of samples, including sample with adverse pregnancy outcomes, which we didn't have here. So that's one thing, to really get deeper into this clinical aspect of the work, get samples and really increase the numbers. But then we also need, I think, to really understand the mechanisms. We have some ideas. For example, we identified this receptor, this anti-inflammatory receptor, DC as being the one used by Crispatus. But then what are the signaling pathways triggered? How does it modulate inflammation exactly? What does it do? What is exactly the ligand? Surface layer protein, sure, but what is the sugar there? Could we convert this sugar then later down the line into anti-inflammatory drugs, for example? So all this is the work that I'm planning to do in the near future. And why is this important? Well, first of all, because there is a boom right now of the development of probiotics for vaginal applications. And there is not really any good ways to select the strains that we are using. As we discussed, there are lots of different. Crispatus is not homogeneous. So to pick a Crispatus strain, how do we choose it? What is the rationale behind that? And we need to understand how this strain interacts with the host and what are the consequences of these interactions to be able to select the best strain. So that's long-term one, the future. What we
Two questions for you, Alexiane, if I could interrupt. Two questions. That's all very fascinating. So, one question for you: is it possible that there is not one Crispatus strain that will work the best for all women? Is it possible that one woman's immune system will actually work better with a different Crispatus strain than another woman's immune system? And in that case, boy, you've got a lot more work ahead of you.
When you look at the receptors of the innate immune system, they can come also in
Yes.
slightly different flavors, so different versions of the genes. And there is no study about that whatsoever in women's health. So we don't know if some women may benefit more from some specific ligands than others. We don't know that. So there could be also, as you are totally right about that, the host component to take into account, and not there may not be a one size suits all solution, but by really understanding better how all these interactions work, we will get closer to have a better design for the probiotics and drugs in general to prevent adverse pregnancy outcomes or women's health complications.
Yeah, absolutely. And I want to give David a chance to jump in here, but very quickly, first, I want to ask, so is the reason that not all the SLPs give off this signal because they are different SLPs? Is that why? Because each SLP, each Crispatus SLP is a little bit different, right? Do you know why some of them do not give off the coding? And is it because they are different organically than others?
So we don't know why some lactobacilli are able to shed more than others. We don't know that yet. What we know is that there are different surface layer proteins that can be found, at least two different. So you could have one version or the other of this surface layer protein. And we also have isolates that are unable to bind to DC sign, so they don't seem to have the sugars. So then you have a whole combination of which SLP do you have, and then which sugars do you have on them. Again, we have only superficial data, I would say, about that. We really need to go down to the composition and the structure of all these sugars and surface layer proteins to understand really what's going on there and what could be the most beneficial interactions and the best strains for further development.
Yeah, and David, I'm still going to give you a chance to jump in here, but Alexiane, I have a question relating to what you just said. And how do you examine the coatings? How can you tell what sugars and proteins are on? This is a very silly question, but is it just like a sophisticated magnifying glass where you look very closely to this bacteria, or is it something else where, you know, maybe dyes are put in, or it's very technological and it's on a big screen? How does this actually work? This minute, tiny, microscopic analysis of these bacteria.
Yeah, well, to be fair, it's actually not very technological at all. I just take
Okay.
my bacteria, use chemicals to strip them. I make them naked. I take their armor away, and
Take their
then we
clothes off.
use
Okay. You're a nudist when
exactly,
it comes to bacteria.
and then we just use simple biochemical techniques such as gel separation to look at the size of it. Then we probe with specific reagents to see which sugars they have. So there are some sophisticated way to look if you want a pretty picture like this pavement like pictures of the surface of the bacteria. You can use not a magnifying glass, an electron cannon, but there are some methods to do that. But really in the lab day to day, we use these very simple biochemical techniques where we just strip the bacteria and analyze what we recover.
And if you didn't have to sleep, Alexiane, if you didn't have to sleep or eat, how long would it take you to analyze fully one Crispatus strain? Because David mentioned earlier there are at least 200 or so or more, a little less than 200, 200 or more, hundreds, he said, at least strains. So we're talking about an encyclopedic dive into each strain. How long does one strain take you to understand? I'm going to do the math and see how long it will take you to understand every strain of Crispatus.
Oh, wow, that's difficult to say. Depends really what you do, but if you want to extensively characterize it, I mean, you
Yeah.
can work for years on a strain, honestly,
Oh my
without
gosh.
sleep.
Oh my gosh. Okay, never
So,
mind. I thought it was going to be like, 12 hours for one strain.
I mean, you
The
can
other factor,
spend,
not cheap, but it's also money. You know, we need some funding to do that type of work.
Well said. Yeah, no, well said. So, David, tell me then your thoughts about the future. You know, there's a lot of work to be done. Where are you going to focus your efforts, and what's the place that needs the most attention?
Yeah, I think these discoveries that Alexiane has really been driving open up a huge range of possibilities as to where to go next. She's already identified some areas and mentioned some areas that she's interested in taking this work forward. In addition to developing, ideal optimized probiotics, and another concept that we're interested in exploring is actually just directly using SLPs or producing a very similar compound that would be able to do the same types of jobs that an SLP would do, and potentially either block a pro-inflammatory ligand. But this requires some really careful knowledge as to when you'd want to block an inflammatory response. We've talked a little bit about this before, where it's still important that the immune system has an opportunity to recognize potentially pathogenic bacteria. We've talked a lot about epithelial cells, but of course there are also immune cells in the mucosal environment of the vagina that are actively hunting for and destroying potentially pathogenic bacteria. And so these types of SLP interactions that we're describing would be involved, certainly manipulating and modulating those types of relationships as well. So some other areas that are quite exciting to think about are coming back to applications, particularly in pregnancy. Most of the data that we're getting now is indicating that having lots of Lactobacillus crispatus, potentially shedding lots of SLPs is a good thing. It's helping turn off inflammation pathways, most likely. So that could be utilized as somewhat of a screening tool, a diagnostic tool, if you like, where we're able to combine with some of the other technology platforms that we've been working on, identify women who are carrying different types of bacteria at the bedside and potentially know whether or not they're producing a lot of SLPs or not so much. And this helps us then to inform a selection, if you like, criteria or a stratification of women who are probably at a heightened risk of preterm birth because of their lack of SLPs, their lack of optimal lactobacillus species. And that's, of course, not going to be all women. We know that probably only around 40% of all preterm birth cases are caused by an infection or an infectious etiology, at least. So that leaves us with still another large proportion of women who are having preterm births for very different reasons. But our sort of approach to this is that if we can at least understand why such a large proportion of women are having a microbial associated preterm birth event, we do have some tools there where we can identify these women more quickly. And now we're starting to get a better idea of what types of interventions and therapeutics we might be able to use to help reshape or flip their microbiome, as we've discussed, and have a better outcome in pregnancy for these women.
Yeah, and David, you said 40% of preterm birth is microbiome related?
Infectious etiology. So of those, a large proportion we think are largely coming from signals from the lower reproductive tract, but there are other infections that can impact other parts of the body, which could also lead to systemic inflammation, for example, that could drive a preterm birth case. But we do think that a large portion of these types of infection-associated preterm births are being driven by a lower reproductive tract colonization pattern. And that's being borne out too in data sets that our colleagues in Stanford and UCLA, particularly Marina Serotto, I know you've had her on the podcast recently, they've been doing some great work looking at how we can take large data sets of vaginal microbiome communities from across the world and find pretty consistent patterns in that data that relate to your risk of preterm birth. So all the data is really pointing towards the vaginal microbiome as being a really important modifier of preterm birth risk. The good thing about that is that we are getting closer to developing the tools we need to be able to modify that risk and improve outcomes for mums and babies overall.
That's fantastic and very exciting. Well said. Alexiane, do you have anything else to add before I thank you both so much for being on the show?
Well, no, I think that covers most of what in terms of future plans, that's most of what we want to do. So
Good. Well, you guys are both doing incredible work, both together and apart. And congratulations on this paper and all of the findings from the paper, from the SLP to the signaling that the SLP does that can be detected in the swab, and all the really exciting future tools that you're going to be working on, from potentially a suppository to a test for preterm birth risk based on the presence of the SLP, and certainly much more that that you guys have up your sleeves. Thank you so much. Congratulations again. And David, you can finally go to sleep now. I know it's past 11 o'clock in Adelaide. So thank you for joining us today. And you too, Alexiane.
I'm never tired to talk about this kind of stuff. And let me just finish by thanking you for giving us the opportunity to talk to you about the study, and most of all, March of Dimes and their supporters for funding this program of work. We really appreciate it.
Absolutely, David. We love to fund you and Alexiane, and I know that the microbiome is a fascinating and one of the favorite topics that we all talk about because it's such a new and exciting field of research. So thank you again for coming on the show, and please come back soon the minute that you have any update on anything that you all are doing, from Crispatus to the inflammation diagnostic test that you're working on separately.
Will do. Thank you.
Thank you.
And Alexiane, let me know how the analysis goes of your Crispatus strains. You've got a lot of work to do ahead of you.
Definitely. Yeah.
Bye-bye. Thank you.
Bye. Thank you.